Hey, welcome back, fellow scientists. You don't get that because you haven't been through the flipped classroom, but that's okay. You'll get to it eventually. That's how I start all my videos because, as we remember from chapter 1, all of us are scientists. All of us are learning about our world, studying God's creation. So, welcome back, fellow scientists. We are going to continue pressing on, learning new information with this remote day. So, as you'll remember from Tuesday. All right, we went through learning objectives. Well, first we took our test, which I'm going to work on grading today. Went through learning objectives, history of the cell. In sixth period, we got through the whole entire history of the cell. Second period, we didn't. So if you were in second period, go ahead, take out your notes. We went through Robert Hooke. We went through Anton von Leeuwenhoek, right? But then we didn't get to, in second period, we didn't get to the cell theory as formalized. So go ahead and write this down, right? So we got the 1665, we got 1675, and then in between 1675 and 1835, that's like 150 years, maybe. I didn't do the math, but you can do the math. Tell me if I'm right or wrong. People are making more observations and publishing more and studying more and doing all sorts of stuff, and people are getting together and talking and blah, 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 blah. And then finally, in this 20-year period right here, they kind of come to a consensus, right? It's kind of like, okay, yeah, we can all agree on this, right? So number one, or letter A, right? All living things are composed of cells. We know that from our 8 characteristics of living things. That's not new news, but go ahead and write it down. Right, letter B, number 2, right, cells are the basic unit of structure and function in living things. So there are things inside a cell. There are smaller things inside a cell. You guys know these: ribosomes, mitochondria, nucleus, different things like that. But what this means is that if you take out mitochondria, it can't live on its own. Right? It'll die. It's no longer going to be functioning. If you take out a ribosome, it can't function on its own. If you take out a nucleus, it can't function on its own. And if it's important enough, like if you just take out one ribosome, that's fine. The cell has plenty of them. If you take out one mitochondria, that's fine. The cell has plenty of them. If you take out one nucleus, then the cell can't function without all of its component parts. So cells, taken together with all of their component parts, are the basic unit of structure and function in living things, right? So they're the smallest thing that can be considered living. And then number 3, letter C, right? New cells are always produced from existing cells. So that's the third tenet of the cell theory. So if you're in second period, make sure that you have written that down. If you're in sixth period, make sure that you have that written down. Hopefully sixth period already has it written down. So we're going to jump right in today for our remote learning day. We're done with number 1, important historical facts about the cell. Number 2, we're going to learn how to use a light microscope. And we're going to start by, let's see if I can get a pen here. Perfect. So I would make this kind of a new section in your notebook, right? So I would title this Observing the Cell. Observing the cell. Perfect. So there's 2 basic types of microscopes that we can use. The first one is this type of microscope right here, which we're going to be using in class. This is called the compound light microscope. Compound light microscope. It has a couple characteristics which I'd like you to memorize. Number 1, we can see living things. See living things. Now, Robert Hooke, or Bob, right, as we referred to him in class, he looked at cork, so obviously dead things, right? So it can also see dead things. But I want to have this here so that we can contrast it with the electron microscope, which can't see living things. Like the specimen has to be dead for the electron microscope to work. But we're not there yet. So that's what I mean by see living things. We can actually observe living cells and living processes using the compound light microscope. Number 2, the maximum magnification due to the properties of lenses and light and stuff like that is 1000x. So max magnification equals 1000x. And then number 3, what's kind of cool is that we can actually use fluorescent dyes or other substances to stain different things inside the living cell. So we can use dyes to stain things in living cells. Perfect. And then here are some examples, right, of some pictures taken using the light microscope. Right, so let's bring this to the front. Yep, good. So this is a plant cell, obviously, right? And we can kind of see the cell wall right here, and we can see all these little green discs in here. So those are chloroplasts, right? And if this was a video, sometimes we can actually see them moving around in the cell. It's actually pretty cool. So here, let's move this down a little bit so I don't have to bring the front every time. This is another image from a light microscope. So these are blood cells, right? So here's our donut-shaped red blood cells, and then here's our white blood cells, which help us to fight infection. Usually when people look at these are dead. These are on a slide. These are prepared. So yeah, but I guess if you wanted to, you could look at living red blood cells. And then here is an example of using fluorescent stains and dyes to stain things inside a living cell. So this kind of green stuff right here, so that's staining the lipids of the cell membrane, right? This red, you can kind of see a little tinge of red, so that's staining the proteins of the cytoskeleton. And then of course this blue or this purple stuff right here, so that's staining the nucleic acids of the nucleus. So those are some examples of how we can use the light microscope and different images that can be produced with the compound light microscope. And we'll learn more about that on Friday when we come back into class. We'll go over parts and pieces, and there's a whole diagram and everything like that. It'll be great. All right. So then number 2, the second type of microscope is the electron microscope. So we'll just use our abbreviation from chapter 2 for electron. Electron microscope. Make sure that you're writing these down in your notebooks, right? When I check your notes for chapter 7, you'll get points for writing this down. So, light microscopes, we have 12 of them. They're fairly compact. You can take them out to the field. You can observe stuff. It's really cool. Electron microscopes, you have to have like a whole room dedicated to an electron microscope. You can see it's a whole big fancy array of tubes and wires and computers and different things like that. So I don't know of a high school in the nation that has an electron microscope. There's probably one or two, but yeah, it's not typical. This is used like high-rel labs here in Spokane, right at the roundabout, right where Future 395 is going to go through. They have electron microscopes, and they use these electron microscopes for parts for the International Space Station to make sure that they scan them and make sure that there's no breaks or cracks or fissures or anything like that. So they make sure that they're good enough to go up into space. So really expensive, really high-tech stuff. So here we go. Contrasting with the light microscope, the specimen must be dead. Specimen must be dead. We'll learn about the process probably on Friday. But basically they have to coat it in gold, and then it goes into a vacuum chamber, and so all the air is sucked out, and if it wasn't dead before, it's dead then. So there are 2 different types of electron microscopes. There's a scanning electron microscope. Scanning electron microscope. And that just looks at the outside of things. So it looks at the outside. Right? And then there's also transmission electron microscope. Transmission electron microscope. And that looks at the inside. Inside of things. So I have some examples here that I'll show you. Number 3, max magnification is 1,000,000x. Because an electron has a smaller wavelength than a photon of light, so we can get higher resolution. So max magnification equals 1,000,000 x, right? So we can barely see viruses. We can see some macromolecules. We can maybe see the tails of the lipid, the hydrophobic tails and the hydrophilic head of the lipid, maybe. possibly. We can definitely see all of the organelles. We can't see like the individual nucleotides of DNA, or we can't see water or anything like that. And then number 4, this is different from the light microscope. Images are produced in black and white. Images are black and white. Perfect. So looking at some images, here we go. Let's get there we go. Perfect. Like that. So this is actually very similar to our first slide that we saw with the light microscope. So this is the inside of a, oh, stop it. Oh, go away. Where is it? Order. There it is. Bring to front. Okay, and then how do I get this? Nope, I don't want to do that. Just go this way. Nope, it's not working. Ah! No, okay. Anyways, we'll just continue. How do I get out of this? Oh, there we go. Aha! Technology will not defeat me. All right, so here's our inside of our chloroplast, right? And we'll learn when we talk about photosynthesis that there's grana and there's thylakoids and there's stroma and stuff like that. But because I told you that this is the inside, what kind of electron microscope are we using here? Yep, that's right, transmission electron microscopy because it's the inside. And this is the image that the computer just kind of churns out or spits out. But scientists recognize that that's boring, and they want to make it a little bit more interesting. So they add some color to it. So this is like taking Photoshop and like adding some. This is not actual dyes. This is not what the computer spits out. This is after we have the image. Then they add false color, and they use some Photoshop and add some color to what they think, just to make the structures a little bit more visible. And a little bit more easy to see, right? So that's a chloroplast. We'll go now to the opposite of chloroplast, right? So this is mitochondria, right? The inside of mitochondria under the scanning electron microscope, or sorry, transmission electron microscope, because it's the inside. And then here's the false colored version, right, of mitochondria, which are photoshopped to make things stand out. Right, just a little bit more. So that's mitochondria, that's the powerhouse of the cell, right? And here, we'll just delete that so we can move on quickly and efficiently. And then, of course, we can do some fun things, right, with the electron microscope. So here is the outside of the head of an ant, which looks pretty cool, like it's from an alien planet or a movie or something like that. So that's obviously a scanning electron microscope because that's just the outside of the specimen, and that's not really blown up too big, right? Because we can see ants without microscopes. We can't see mitochondria without microscopes. So it's kind of fun to look at things in depth, even if we can see them without the aid of a microscope. And then, of course, we can have some fun false coloring an ant, and maybe have it as a stand-in for an alien on a sci-fi movie or something like that. But there's an ant after it's been photoshopped. After it's been electron microscopied, and then after it's been photoshopped and stuff like that. So hopefully today we learned a little bit about microscopes. Like I said, we'll be doing a lot more with microscopes in class, but I just wanted to use this time wisely. Make sure that you have all of the history of the cell written down in your notes, and make sure that you have all of the observing the cell and all the parts of the microscope written down in your notes. This has been Mr. Leo. Thanks so much for watching, and I will see you on the flip side.